I think it's fair to say you guys are in the climate change business. You're trying to mitigate greenhouse gas emissions. Going forward is going to be increasingly emphasized across nations. And if you're a young person and you have an interest in ruminant biology and ruminants in general and you're in nutrition, let me recommend you look at this and read these articles, and it'll give you a start in this area of science. This is an area of science that is going to continue to grow, improving the efficiency of ruminant food production and also minimizing their impact on the environment. Hello, and welcome to this special episode of Dairy Digressions, the podcast of the American Dairy Science Association, the Journal of Dairy Science, and JDS Communications. I'm Matt Lucy, former editor-in-chief of the Journal of Dairy Science and JDS Communications, and fellow of the American Dairy Science Association. I will be your host. Dairy Digressions is where you get an inside look at the science and scientists that are making headlines both inside and outside the dairy industry. We explore the cutting-edge research that creates knowledge and drives innovation in the production and processing of milk for human consumption. Mark your calendars, the 2025 ADSA annual meeting happening on June 22nd to 26th in Louisville, Kentucky. We are currently calling for abstracts between now and February 12th. Abstract submissions are open, so showcase your work to the global dairy science community. And also remember that our abstracts are published in a supplement to the Journal of Dairy Science. So get your work out there in abstract form and join us in Louisville, Kentucky. Today, we have a special episode, and we are going to discuss a special issue, feed additives for methane mitigation, that is coming in January 2025 in the Journal of Dairy Science. We have with us today two of the key authors of these manuscripts. Remember that these articles are open access. And what open access means is that if you are listening to this podcast, you can follow the links and get to this special issue and learn everything that these guys have put together for you. We also have two other special issues that I'll highlight at this time, one devoted on the highly pathogenic avian influenza, H5N1, and another on dairy behavior in animals. Now let's get to the podcast today. So first of all, we have with us André Banik, who's a researcher in animal nutrition on Wageningen University. And also with us today is David Yanez Ruz. He's a senior scientist in the Spanish National Research Council, and his experiment station is located south of the city of Granada. Welcome, gentlemen.
Thank you.
Hello, thank you.
These two gentlemen, I'm not sure, gentlemen, first of all, can you just explain, we want to talk a little bit about this special issue. It contains an introduction by a gentleman, Dr. Cruiser, as well as 6 individual articles. And this work has arose from a project, a flagship project entitled Feed and Nutrition Network of the Livestock Research Group of the Global Research Alliance on Agricultural Greenhouse Gases. So I'm going to let one of you tackle this whole, what is this network? What did it come from? Explain about that, first of all. You decide who's going to tackle this.
David is the chair, the current chair.
Okay,
okay, David's the chair.
Yeah. Well, you were the past chair as well. But let's start with the big umbrella. So the Global Research Alliance for Agricultural Greenhouse Gases is an international initiative in which 68 countries collaborate together to put science into trying to reduce greenhouse gases from agriculture in general. And we did that under that big umbrella. Then we have different research networks, one of them research groups, one of them being the Livestock Research Group. And within that group, we have different specific research networks, and one of them is the Feed and Nutrition Network. There are others dealing with breeding, microbial genomics, etc. But ours is dealing with any feeding strategy that can contribute to reduce greenhouse gases emissions. And this network started to operate in 2011, 2012. in which different countries joined, and we had different projects that have produced several publications, several models and prediction equations that have been also incorporated by the IPCC into the prediction models. And in the last 2 years, we have been running this flagship project within the network that aimed to produce guidelines, good practice recommendations to develop feed additives with the objective of reducing enteric methane emissions.
So does this global alliance, I'm trying to think of how I would, okay, the word alliance is in the title of this, so we'll call it a global alliance. Do they provide funding?
No.
No funding.
No. It's a collaboration of scientists, at least in a network it is. There will be some funding by governments, particularly the New Zealand government, who started up all this Global Research Alliance. So they fund the administration and the hosts and the chairs and all those kind of things.
Mm-hmm.
But the networks, it's all on in-kind basis. So it's just a collaboration of researchers. And every time, if we want to do something together with all these researchers, which David just explained, we need either a project or we need to find money, but they help us in finding some, as they also did for this flagship project.
So would you, and it's gonna take a few minutes for our listeners for us to peel away a few layers, but before we peel away further, can you explain, is the European Union funding your research or supporting your research, or is it your nations per se? Your nations or the European Union, or a combination of both?
Well, it depends on the different contributors basically contribute with time and experience from their own labs.
Mm-hmm.
And in some projects, we have received money from the European Commission, but from other international entities. And in this particular project, a flagship project, we received funding from the Global Dairy Platform. And they funded 2 years postdoc contract to help and to assist Andrea and I in coordinating the project. But the rest of the effort, all the authors that contributed to the papers, that was their time, which was in-kind contribution within their activity. So there was no further funding to help with this. But it is very difficult as well to provide funding for such a long and extensive work in which 65, I think we were in total, authors were contributed on different papers, because we had to find a time and we had to collaborate with each other at different periods during the 2 years. So it's a kind of difficult project to fund, because it's not like, if you need some specific amount of money then to run experiments, then here's the money. It's just the time that researchers need to sit down and discuss how to approach these guidelines.
Mm-hmm.
It's on top of the other work, which is funded normally. This is just something on top, which we think is relevant because we need to collaborate. We need to find common grounds because all the research groups, they do similar types of work, of course. And we need this kind of network to discuss, come together, and also to deliver these kind of projects. But for the senior scientists like David and me, it's in kind. There's no additional money unless people in our own organization pay us for a few hours.
Mm-hmm.
But it's basically in kind. But we got a lot of help from the funding of this postdoc. The postdoc helped us quite a lot in taking work away from us.
This is interesting. So, and so let's back up for our listeners a little bit. Okay. So these two guys are dairy nutritionists, and you're interested in methane mitigation. And let's remind our listeners that and I would be curious what the real number is, but so methane is one of the greenhouse gases, fair? And enteric methane comes from the rumen of the cow. We know it's produced by, I guess, they're bacteria, methanogen. I'm already wrong here because David's like, well, okay, we'll get to that in a second. But methane comes out of the cow herself. It also comes out of the manure pile. That's another place, but we're not going to do that.
Correct.
But there's different, and I think in total, it depends on how they do the calculations, but of total greenhouse gas emissions, total greenhouse gas, not methane, is it fair to say that ruminants contribute around 8% of the total greenhouse gas emissions? Is that close?
It depends on the nation, but on a global level, I don't know the figure by head myself, but it's in this order of magnitude, yes.
Okay. And I will cut to the chase here a little bit. You guys are nutritionists. You're specifically interested in what can we feed the cow to perhaps change the amount of methane, enteric methane, that comes out of the cow. Is that fair?
Yeah.
And that's one option, is the way we feed a cow. The other option, of course, is breeding and genetics. And there are individuals who are interested in, maybe through genetic selection, identifying animals that might have less enteric methane. And for those interested in that, in this podcast, we've talked to Filippo Miglio from Canada, Donna Berry from Ireland, Christine Bays also from Canada about this. So we have discussed this genetic aspect. We've spoke with Frank Mitlerner from UC Davis in all aspects of methane, including digesters. But these individuals we're talking about today, and this special issue, has to do with feeding cows in a unique way. Okay. What you're describing, gentlemen, to me with your alliance sounds a little bit like a scientific society. Don't we form scientific societies and we do work for them at no pay and all that other stuff? Is it a little like a scientific society with an interest in methane and feeding strategies? Is that fair?
It is, yeah, in a way, it's a society. Yeah, it's a network, a group of scientists that been collaborating for quite a few years now and co-benefit in that interaction, and sometimes with common funding, and sometimes with just the interaction itself through collaboration projects like this one. But if I can add something about the nutrition methane link, I think it's important to say that dairy farmers, that they don't feed cows, they feed microbes in the rumen, and then the microbes ferment the feed, and then they feed the ruminant, the cow. And part of that process is when methane is produced by microbes that are similar to bacteria. They are called archaea, and that's our part of our interest in how we can modulate that fermentation to reduce as much as possible methane without affecting or even improving the efficiency of that fermentation.
Yeah, I think for our listeners, you have to understand that we are dealing with living things that produce human food. And in a living things that produce human food in a commodity market. And what I mean by commodity market is trades at break even. And so it's important that when we work with this living thing producing human food, that we don't mess her up, for example, and make it so she no longer produces enough milk. But we also have concerns of not messing up the consumers that consume that milk and maintaining their trust. This is a serious landscape that we work in here. And I think you'll come to find, as we talk to these two gentlemen, that they are individuals who can care sincerely about the production of food. And you obviously care in a global manner. And you care about the production of food in a manner that maintains a healthy earth and a healthy world that we live in, which is, can there be no higher calling? I mean, it's so easy to go to work every day when you feel like, you know what, the reason I do this is because I want a healthy planet. It's interesting, isn't it? And I want to produce food for a healthy population, and we never have to feel bad about that, or anything like that. I'm surprised you could find 68 member countries. Oh my, are there that many countries with animal scientists in them that are 68? Dig deep, huh? 68 countries.
Yeah, but in principle, we are global, and it means as a network, we are quite open to any country, basically. If there's a good representative or a person eager to join the network and who can contribute, they are welcome to join.
Yeah.
And that's how you get to 68 countries. You start counting them on the globe. There are many. There's still quite big regions that are not that active in the network. We would like to have them as well, like Africa and those kind of regions. That's more difficult.
Yes.
But the more intensive dairy production system countries, they are all involved in the network, all of them.
I am curious. I've always, for the Society of the American Dairy Science Association, we don't have a lot of members from Africa. We have some members from the Middle East. We have not a lot of research comes in from India. Do you have many people from India in your organization, the country of India?
I'm not certain. I think
that's a good question. I think, as Andres said, that the Global Research Alliance initially started with a few countries that were promoting the initiative, mainly New Zealand and some European countries. And then, yeah, that kept widening and including some countries from Africa and lately from Asia. But Asia is not. India is not currently, I think, a member. China is a member. Malaysia, Indonesia, but there are still big gaps in some countries that are not part of the Global Research Alliance. This doesn't mean that we don't collaborate with researchers in those countries, but this initiative is a governmental initiative, so the governments need to decide to become part of the Global Research Alliance, and then the scientists from those countries can come into the whole network.
Yeah, I ask about India because I think there's more dairy cows in India than any other place in the world. I mean, there's a lot of cows in India, and it's something I'm going to have to get somebody from India on the podcast and just sort of ask them about all those cows there and try to understand that a little better. And I'm always interested when I read things about India and the dairy industry there. Hey, I wanted to ask you. So the introduction of the special issue. So there's a 2 or 3 page introduction written by a Dr. Kreuzer from Switzerland. Can one of you explain why was this individual selected to write the introduction and who this person was, so that our listeners can understand Dr. Kreuzer from the Institute of Agricultural Science in Lindau, Switzerland?
Well, he started this feed and nutrition network. In the beginning of the Global Research Alliance, there were a few networks, but one on nutrition was missing. So Dr. Kreuzer, he felt that was a gap because nutrition is so important and so much dictating what is happening in the dairy sector and in the beef sector. He thought there should be a nutrition network as well. So one was founded by him, basically. He invited all of us to Switzerland and we had a good meetings over there. And then the network started. And fortunately, Alex Christoph from the US, he was involved as well. He was able to quite soon after that first meeting get a joint programming initiative, they call it. It's collaborative research across many countries. And also the US was eligible to join in those projects. And Alex Christoph started a project named Global Network, where we would share data sets that all these research organizations have to make best use of all the data that are there around.
Alex is the author of so there are a total of 6 papers in this. And again, I would like to emphasize that this is going to be the Bible going forward in terms of how to approach studies on feed additives to mitigate methane. And Alex Hirshoff, he's from Penn State Animal Science. He wrote recommendations for testing and methane mitigation. Recommendations, experimental design. How do you design an experiment? What should be included? What should not be included? So different statistical approaches. It's quite an article. Let's just put it that way, And if you're interested in dipping a toe into this is you would probably start by reading this article and trying to understand what that is. And so I just want to ask you, I'm going to go past a lot of the statistical and experimental design stuff. I just want to ask you, it seems like when I listen, or when I was the editor-in-chief for Journal of Dairy Science, no one could really agree on the best way to measure the methane produced by an animal. Okay, what is the best way and the most believable way? And it was always this argument, okay, your system you're using is wrong or right. And Alex really kind of goes through all sorts of different methodology. Is there any consensus on this amongst the methane community of what is let's just start with maybe an animal on a TMR, a confinement animal, and contrast that maybe with the best, most reliable method to measure methane in a grass-based animal out in the field. Okay, because if you can't accurately measure it, you're nowhere. Any thoughts on that?
Well, yes, there's been long discussions on this topic, and we have, as part of that project that Andre mentioned, that was led by Alex, published a few papers on trying to review the different technologies available, which have been evolved in the last year. So this is a dynamic discussion. So it's not something that is set forever. But I think, thank you to that review paper and other collaborations. There is a a good consensus now on what is the gold standard on how to measure methane on an animal that is fed a TMR on a confined system, which is basically using chambers. So the animals are kept in a kind of enclosed environment where you can control how much the animal eats, and you can continuously record methane, hydrogen, and CO2. So you have a good recording of the whole exchange of gases. But then technology has also developed other tools that have been used by Alex and other colleagues, like GreenFeed, and so shorter term measurement technologies that can allow to have the animals standard farming conditions or even grazing conditions. And those technologies have been improved quite substantially in the last years. And the paper also identifies the needs for future research, so the potential improvements are needed to get those systems to really measure methane as accurate as possible. But yeah, I think there is a general consensus of what is good practice and what is not in terms of measure methane. But the first point is really important. If we cannot measure, you cannot improve what you cannot measure. So if we can measure methane properly, then that's the first step. And that manuscript led by Alex is I think it's the largest in the set of the 6 papers.
Mm-hmm.
And provides a very useful recommendations on how to design those trials, what things need to be considered depending on the system that you're using, and then what other options you have depending on the different conditions that your system operate.
Yeah, I think what's really helpful is finally, like, we really struggle like this in some of the reproductive research and some of the definitions without sort of a flagship paper. There's just arguments that go on and on and on. And Alex is so well respected across the dairy community. It's really helpful that he actually put a knife in the sand or whatever, just said, okay, this is what I think. Is it true? Okay, so we talked about the fact that if you've got a cow on a TMR, you can stick her head in the box and sort of measure the methane coming off her. But is it also true that the grazing animals is it a lot harder to sort of assess methane in a grazing animal than a confinement head in the box kind of animal? Or is that true?
It is more challenging because the animal is not confined, and you as a researcher have less control on what the animal is doing. But you can use the same green feeds also outside in the field. As long as you check carefully how the animals behave, whether they visit the measurement device, the green feed, and if the animals actually do that in time, and if it's well spread across the day, and you can do all these data checks, then you have good measurements also out there in the field.
Alex brings up 3-NOP, and I'm going to use that as I'm going to use 3-NOP as, and I'm going to let one of you describe exactly what 3-NOP is, but I'm going to use 3-NOP sort of as a test case for a couple of things that Alex brings up in his paper. And he brings up animal health and the need for understanding animal health. He brings up animal quality, not animals, so it's the product, the quality of the milk produced and the safety. I think he's probably talking about human health safety here. Can one of you just take that? Okay, so I think, am I incorrect when I say that 3-NOP, people are going to start hearing a lot about 3-NOP. Is that one of the first major rumen modifiers to be put out there that's currently going to be fed to dairy cows to reduce methane production? Is that the first one, for example? Is this true or not?
In terms of reducing methane production,
yes,
I would say yes. 3-nitroxypropanol was first tested in vivo in our lab in Granada in sheep before it went into cows. And that was 2011, I think, 2010, 2011. So in the last 13, 14 years, there's been quite a lot of research undertaken in I would say all continents in many different conditions. I don't know how many papers have been published now on testing 3-NOP in vitro, in vivo, short, long term, checking all rumen fermentation parameters, milk production, milk quality, etc. And now going into longer term trials. So checking that, if you go through 1-2 lactations, what happens to animals. It's been the most studied molecule in terms of reducing methane emissions in ruminants, and it's been approved for use and authorized in, I think it's over 60 countries now, and including the whole European Union.
I'm going to unpack that a little bit. So I think when I read, you get about a 30 to 40% reduction in methane if you feed 3-NOP. Yeah. Okay. So that's the correct number, which is a big hit. That's
On
those are
average,
big numbers. Okay. Okay. So in America, I don't know, probably Europe, there was all this excitement over feeding seaweed to cows to reduce methane. How does 3-NOP compare? And I know there's issues with seaweed, and we'll probably talk a little bit about that in a second, but how does it compare to feeding cows seaweed? Just in terms of a biological effect on methane production. Same, less, more, where are we at?
I think the order of magnitude of reduction that you can achieve, it's also quite big with seaweed. If it contains the active compound that is inhibiting the methanogens in the rumen, this bromoform or bromoform-alike compounds, they inhibit the methanogens just as 3-NOP does. The concern always is, okay, but what does it do with the cow? What is happening with the metabolites? How are they ending up in the animal? Is anything coming into milk? Is it safe to use? All these things have been done for 3-NOP already with these EFSA trials. That's for the regulatory body of the European Union. They had to show all this information already for the 3-NOP.
Okay. So
with the seaweed, it's a bit strange. If it's seaweed, you are allowed to feed it because it's blue. It's a feed material that you can just use. If it would be the compound isolated, you take the bromoform out and you feed bromoform to a cow, then suddenly you have to, of course, investigate all the safety issues because you feed a compound. So with the seaweed, it's sometimes a bit, is it seaweed or are we talking about bromoform?
And seaweed is an interesting one. I think somewhere in the special issue, they talk about the fact that seaweed has high concentrations of iodine in it, and feeding
also
high concentrations of iodine, that has implications for the health of the cow. And biologically, of course, as you two know, you need iodine in your diet for thyroid function, but too much iodine in your diet is not healthy. Okay. And so you need a certain functional level of that. So it's not just so simple as well, just seaweed is safe and seaweed is safe, but there's more to the seaweed story than just whatever, and it's a new feed ingredient for cows. What about ionophores? Ionophores also inhibit methane. Do they inhibit methane at the same level as 3-NOP? Or are they just not even on the same scale in ionophore?
They are not on the same scale. To compare them, I think it's important to maybe to go to one of the papers of the special issue, which is the mode of action.
Yes.
There are different ways that a molecule, a compound can reduce methane.
Yes.
One is directly inhibiting the activity of the microbes that produce methane, methanogens, and that's the case for 3-NOP or bromoform in seaweed. But others can modify the overall microbial fermentation and reduce the production of some of the substrates like hydrogen that is used by the methanogens. And that's the case of the ionophore. So there are some modifications in the overall fermentation that can result in less hydrogen being produced and then less methane at the end of the pathway. But it's not a direct mode of action. So it's not targeting directly the microbes that produce methane. And the reduction is not 30, 40%. And it's very variable because it depends on the diet as well. It depends on the substrate that's being fermented. With 3-NOP, for instance, you know that you are within a very narrow range of mitigation because it's very direct action into the methanogens. It can vary depending on the diet, but the variation is really tiny compared to other compounds like ionophores, where you can have no effect or 10%, for instance, but it doesn't go as high as with direct methane inhibitors. So the mode of action is a very important factor that determines whether a compound is very effective or not.
Yeah, so you're speaking of the paper by Alejandro Balanch, and we're going to talk about that paper in just a second because it's an outstanding paper. Outstanding. All the papers are phenomenal in here. I had a question, as a consumer, I actually looked at this molecule, the 3-NOP, and I know enough biochemistry to go, looks pretty safe. It's not that complicated. I mean, when I was working for Monsanto and we were working on bovine somatotropin. There's a lot to that molecule, and there's a lot of moving parts. Now, this molecule, to me, this 3-NOP, it looks like, I don't know, some propionate and some, I don't know, nitrogen thing. I don't know. It doesn't look that complicated. Is that fair? Is that why it's considered to be so safe across so many different regulatory agencies? Is, you know, you
This
feed
benefit,
it,
I think, yeah, of the compound, yeah.
you
Because the
can
compound,
basically,
it blocks the methanogens, but as soon as it blocking this enzyme in the methanogens, the compound disintegrates and it becomes nitrate and some propanediol compound. And the propanediol, those type of compounds are already in our silages. So it ends up into a metabolite that's not really that much different from what cows feed anyway with corn silage.
right. And so I think that's important for any consumers listening, that
Yes,
this is a very simple molecule. It's
yes.
not that heavy a lift to kind of understand how it's metabolized, and it's quickly metabolized to other molecules that are just present in the rumen. And it's not a scary chemical. It's not a, you know, it's you know, for so many regulatory agencies, including the FDA in the United States and the European Union. European Union is usually tougher than the Americans, I think, for some of these things. For them to green light this thing, it's got to be quite safe. And then I don't think we feel, based on Alex's, or that there is major effect on milk. A lot of the carbons that are present in this molecule, I think, end up in milk as milk sugar. I think that's where they sort of end up. Isn't that true?
Yeah, but yeah, that's true. Once is initially degraded, then the nitrate goes one way, and then the carbon chain goes the other way.
Yeah.
But we're talking about 60 milligrams per kilo of diet. So we're talking about tiny amount. So once is degraded compared to a cow that eats 25, 30 kilos of dry matter, it's nothing. But yeah, it doesn't, because it degrades so quickly, it doesn't bring any concern. I invite one of the papers as well in the special issue is dealing with the different regulatory frameworks that are now in place in different regions of the world. And the ones in the European Union probably is the one that started first to have a well-defined guidelines to go through a dossier for authorization of these compounds, and the dossier of and the evaluation of 3-NOP is publicly available. So I invite people to take a look at it and see the information out there. That's publicly available. The results of the trials conducted by different research teams with DSM, and you can see, yeah, what happens with this compound when it's put in the rumen and quickly degraded, and the different trials that were testing the fate of that compound and whether any of the organs of the animals were affected when a much higher dose was used. And as you said, the EFSA regulation is quite tough, and it took, I think, 7 years to get through the whole process. So yeah,
Yeah.
it's just a matter of taking a look at the information that is available to see that, yeah, it is a tough process to make sure that a product that can be used in dairy nutrition is safe, and this is one of the cases, I think.
What David is talking about, the fact is that anytime you have a new feed additive that's not generally regarded as safe, that's called GRAS. You know, anytime you have a new feed additive, you've got to do things like tox studies. And a tox study is basically, you've got to feed it at your recommended dose, but, you know, maybe 3 to 5 times that dose to make sure there's no effect on the animal. And so this molecule, this 3-NOP, just basically passed all those hurdles. I don't think there's any evidence of any toxic effects on a cow or a human. And so I think this one really looks like a really good molecule. Now, I have a question for you guys. Now, in the European Union, it might be different from in the United States, and maybe you can touch on carbon credits and the carbon credit market. I don't know that you have a carbon credit market in the European Union, but in America, we have this carbon credit market. So and in America, there's no reason for a dairyman to feed this. I don't think they take any, you know, the politicians make rules that you got to reduce this and that and the other thing. But why would a dairyman feed it in America? And why would a dairyman feed it in the European Union? And what is the incentive for a dairyman to purchase 3-NOP and put it in the cow ration? Why should, other than it makes me feel better?
It really depends on the country and the scheme that in different countries is adopted. In the European Union, it's not one scheme for all EU nations. It depends on what politicians in Belgium, Denmark, or the Netherlands decide on.
Mm-hmm.
So in Denmark, I'm not sure what status the law has, but they want to have it in their laws that methane has to be reduced. If it's a law, then farmers have to do it. They simply have to do it, and it has to be compensated for. In Belgium, they also had a kind of compensation scheme that farmers actually get paid for using the compound. In the Netherlands, it's more the dairy sector itself that starts to stimulate the application of the compound because they have to comply with EU legislation that big companies, they have to show that they are doing their best in cutting down greenhouse gas emissions. So it's a
Mm-hmm.
kind of stimulation for the dairy sector to show that they go down in greenhouse gas emissions per liter of milk. And that's actually where the stimulus is coming from in the Netherlands. So
Okay.
farmers literally get compensated for using Bovaer into their diets.
Okay. And so I think in America it might be I'm not sure every state might have different laws. Part of the
Probably.
compensation though I think is through the carbon credit market. I think I might be wrong, but I think that's part of it is if I can reduce my carbon, I can sell it. Does that sound right? Does that sound right at
Yeah, I
all?
think so. Yes, yes, it will, and it is accounted for. So it's already applied in several countries and also in the farm assessment methodology that farmers fill in what they are doing.
Mm-hmm.
And then the assessment tool delivers a kind of greenhouse gas emission per liter of milk, which is being used by the dairy sector for trading.
Mm-hmm.
And the effect of nitrate, for example, but also of 3-NOP is already introduced in those
Yeah.
assessment tools.
Right.
So it's
Yeah. So
accounted for. So
yeah.
also the dairy sector can do their calculations.
We're going to move to a different part of this review, but I would like to ask a question. I remember that one of the reasons that justification for ionophores is that if you can reduce methane production in a cow, that will basically make her more efficient because methane is an energetic loss to the cow. Does 3-NOP make the cow more energetically efficient? Is there any evidence that they're energetically more efficient because they're not losing methane?
That's a very interesting question.
Yeah, I
Well,
can.
yeah.
Sorry.
No, you can go, David.
That's fine. Well, the initial hypothesis is just because methane represents a loss of energy for the ruminant. If you reduce methane emissions, then there is theoretically more energy available for the animal for either milk production, growing, maintenance, etc. But the truth is that at the level of reduction that we have achieved, 35%, there is no clear benefit in terms of energy metabolism. And the reason for that is because basically we are blocking methane production, but we are not making use of the excess hydrogen that is not used for methane. And we are not the rumen is not diverting that hydrogen towards other metabolic routes and produce other metabolites that the animal can use. There is some research going on now in trying to combine additives that can solve that problem. So by basically making use of that excess of hydrogen and therefore providing more energy to the animal. But so far, it's just very initial research, I would say. But yeah, the theory is that we could make use of that methane, but in practice, unfortunately, it's not the case.
Can I add a little on that?
Yes, yes.
That it's also difficult to demonstrate with the trials that are normally being done. They are
Mm-hmm.
not hugely long. It's difficult to demonstrate a small positive effect to get it significant. And if it's not significant, then we as researchers, we are not allowed to say it works
Mm-hmm.
or you have a benefit. We ourselves did one trial for 1 year, and in that trial, we did see a benefit.
Mm-hmm.
But that's only one trial. So there should be more long-term trials, I guess, to demonstrate whether this consistently happens, yes or no. But personally, I have a feeling the hydrogen is not coming out of the cow. It's not being
Mm-hmm.
used to make methane.
Mm-hmm.
Some is coming out of the cow a bit more, but most of the hydrogen is somehow disappearing. It ends up in the cow somewhere. So I would suspect the same as you said. It has to go somewhere, so it has to become energy. But it's
Yeah, but
difficult to demonstrate, to prove literally that the cow benefits. That's the big
yeah,
problem at the moment.
and the truth is that we are kind of fighting against a machinery that has been evolving for so many years. So ruminants were present thousands of years ago in our planet, and that ecosystem has been adapting to the different conditions to be able to digest forage and develop their own mechanisms and the mix of microbes. And then all of a sudden we bring something that knocks out one particular pathway, reduces methane production by 35%. And then the ecosystem goes a bit crazy, like, ooh, what's this? So we just in a few years, we have changed something that has been evolving for so many years. So I guess it will take a bit longer to get a full benefit of that change, I imagine.
It's interesting to see how this all goes. Microorganisms are rapidly evolving species, right? And so it'd be interesting. Although I think you've done long-term trials, and you feel pretty confident that the methane mitigation of 3-NOP doesn't go away after a few weeks or something like that. It seems to be effective, and so more on that. So Jan Dijkstra from Wageningen also wrote an article for this on sort of modeling of the additives on their methane emission, and we're going to be bad, and it's another outstanding addition, but he's talking all about how you mathematically model these different effects, and for those interesting in a really nice tutorial on modeling, and he really goes through the different types of models and how they would apply. It's outstanding, but we're going to jump ahead. Don't tell Jan. Okay. He's going to be mad at me. Okay. But he's already been on the podcast. We're going to jump ahead.
That's true.
And we're going to talk about this. We mentioned earlier, Alejandro Balanche. I don't know if I'm pronouncing that right, from Spain. So he must be David, is he a colleague of yours who wrote this?
He's a colleague of mine. Yeah. He worked in our lab for 6 years and we still work together. Yeah. He now works in University of Zaragoza in North Spain. So he's a very, very good friend and a very good scientist.
And how do you pronounce his last name?
Balanche.
Okay. And he's done I think one of the things that's really sort of excellent about his article is in his first figure, he actually goes through sort of the biochemical pathways and the mechanisms of action. This is again a fantastic tutorial for just kind of understanding how you might approach this or think about methane mitigation in ruminants. And with that knowledge, I'd like to jump ahead to the paper by Zoe Dermick, in which she's talking about recommendations for identification and selection of new compounds. Okay, this is 3-NOP. The 3-NOP story is over, guys. I mean, it's done, right? I mean, it's going to get used. It works effectively. What's the next great 3-NOP? And will it work? Will it complement 3-NOP? And I think Alex had a little bit about that. Will it complement 3-NOP? Can we get further methane mitigation? Okay, so let's just start with that first question. Can we reduce methane further, or are we at the biological limit of methane reduction in ruminants?
I think we can further reduce methane without obviously compromising overall rumen fermentation and therefore the animal's health and milk production. You mentioned this paper by Zoe Durmick, in which it describes the first steps in trying to identify potential candidates, and that was the case of 3-NOP many years ago. And in the old times, we were testing products on extracts from different plant compounds, not randomly, but just testing lots of products in vitro in our labs. Now we have the capacity to develop or to use computational models to find potential candidates that can target a specific metabolic routes or specific enzymes, such as methyl coenzyme are in methanogens. So by knowing the specific pathways we want to target, then we can try to find by different softwares what molecules can do the job. And then obviously you need to go further and then test them in vitro or in pure cultures with the cells of methanogen first, and then with rumen fluid, etc. As these computational techniques develop, then I think we are able to go further in trying to find other molecules. And that's one way. The other way, and I think we mentioned that previously, is trying to combine compounds that have different mode of action by reducing methane and see if by adding something that reduces 25 with something that reduces 10%, you can get to 35 or even more, because they can be synergistic, so they help each other. So that's the kind of work that is now ongoing in different labs, and not only trying to identify new molecules or modified molecules, but also what kind of combinations can be used to go a bit further in the reduction, of course, without really affecting the microbial fermentation.
That was an excellent description of this article. It brings me back again to my days when I used to work for Monsanto, and we had a biopharmaceutical company that we owned at that time. And, part of the strategy in identifying new chemicals at that time, now this was 30 years ago, was to basically, we called it the Whitman sampler approach. And if you don't know chocolates, you won't know what a Whitman sampler is. But a Whitman sampler is a box of chocolates that contains lots of different varieties of chocolate. And the Whitman sampler approach was that you just basically developed a bioassay, and then you hit it with every chemical you could find, either isolated from plants or bacteria or wherever, and just ask the question, which one of these chemical pools would successfully give me the result I want? And then you pursued that. And I think this is a little bit what he's talking about with this empirical approach. In other words, don't make too many assumptions about what's going to work. In this paper, he talks about this empirical approach. Don't make too many assumptions about what's going to work. Just develop a bioassay or an assay, some sort of assay, and just hit it with lots of different chemicals across the plant and animal world, because you can't really predict. And of course, if we think about one of the greatest examples of that, it might be rapamycin. And rapamycin, of course, was isolated from, I think it was from fungi from Easter Island. And of course, it became one of the most important immunosuppressant molecules of our time. But now it's a different time. And what David brings up is the fact that we now have particularly with AlphaFold and all the new things, we have the capacity to understand the three-dimensional structure of enzymes that are involved in these pathways. And that's, AlphaFold, they just awarded Nobel Prize in AlphaFold. And now based on molecular modeling, you understand where the active site of that enzyme is and how do we block that active site. So I don't know that we need to do the Whitman sampler approach any longer. The future is basically targeting certain elements of those enzymes. So is there any, given that we're in this new age, has the new age produced any? What's the next great molecule? Or you guys are too busy working on 3-NOP to work on the next great molecule? Is there another molecule?
I think a lot of work is on 3-NOP and bromoforms. Yes.
I think it looks like the way you guys are looking at me, you guys own, biotech companies, and you just don't want to talk. I mean, come on, there's got to be something else out there.
Well, I think, of course, there is a huge surge using this new power that we have, technology power, to look for other new molecules, and that's fascinating. But at the same time, there are also challenges with the molecules that we know work that need to be still addressed and that keep us busy as well, in terms of how we deliver, for instance, 3-NOP or any other molecules to grazing systems. Yeah. Because we need to combine or synchronize the delivery of the molecules with the fermentation of the forage, with the diet. So methanogens are inactivated. So if you don't have that synchronization, for instance, in grazing animals, when the animal is grazing, 3-NOP cannot enter the rumen because you cannot mix it with the grass. Then how do you do that? So that's one of the challenges that with not only 3-NOP, any other molecule that still needs to be developed. But of course, there is a huge research effort in trying to develop new molecules. But God knows how many companies are running different trials in that space.
Do you think that you guys have been in the 3-NOP world for a while, and you guys are nutritionists, and we have a lot of young people that listen to this podcast? Okay, so I'm going to ask you both. I'll start with Andre. I don't know how old you are, but you're old enough.
62.
62. Do you have any advice for young people that, they're listening to you guys thinking to themselves, wow, these guys have spent their whole lives working on trying to mitigate methane. Do you have any advice in terms of what classes they should take, or what approach would you take? What do you wish you had in your toolkit as a 62-year-old that you could have now, but you're sure as heck not going to go back and learn that or do that? Anything in your science?
What I'm missing now, you mean?
Yeah.
What I should have done when I was young.
Yeah.
Yeah, that's difficult to say. It's on the molecular sciences, for example. I'm a bit less involved in that. It's a bit too complicated for me. like designs of molecules, locking an enzyme, things like that. That's too far away from me personally. But it's really intriguing, and also the whole software that is available nowadays to explore that area. That's a world in itself, and it's really powerful. We just discussed it. It's being done, but that's a different type of science almost. So I feel a bit sorry that I missed that path. All the other paths, I'm fine so far. I never imagined that all the things that I did when I was younger, the classes that I received, that I actually would use it as a 62-year-old. But all my high school classes, all that stuff, it's all coming back. And I needed it, all of it. So I would just advise young people have this perseverance, even though you don't see the use of learning all of that. If you follow this path of doing research, I think in the end it will come together and you will need it. You will need it. But it's difficult to find what you really want to do, where your ambitions are, in what direction you want to go as a researcher. I found personally that the most difficult decision.
David, do you
What
have
to
any?
choose? Are you going for breeding or
Yeah.
nutrition or physiology or
Yeah.
health?
What do you think, David? Something that would help you?
Well, I think Andrea and I have been very lucky to witness or also to be part of this development of feed additives that in 20 years ago or 30 years ago was based on wet microbiology and just more lab work, and now is combining all those disciplines. I think, where I missed that it would have been nice to have many years ago is that interaction between biochemistry, microbiology, physics, nutrition, that now it's happening. And if we had that level of interaction 20 or 30 years ago, I think we probably would have made much more progress. But yeah, that's how science works. And yeah, we know now that by interacting with people that can add a bit more knowledge to our knowledge, and then help to develop any product. But if you're asking about advice as well to young people about I think regardless of the level of technology, and I think in our work, curiosity, consistency, and critical thinking are very important. And yeah, sometimes you're lucky, but sometimes you're not. But this is like training for a marathon or for an Ironman. If you have consistency and you stick to and you go through a good program, you get somewhere and you succeed.
You guys have said it really well. I think this is me, but yeah, I was born in 1960 and I'm 65. No, not yet. Almost 64, 65. But, DNA was discovered and was reported on in 1954, the structure of DNA. And I always say this is probably overstated, but has there been a greater 60 or 70 years in science? I mean, maybe every 60 or 70 years of science is greater than the last. But just think of, DNA in 1954. Oh, that's an interesting molecule. And look where we are today, and I think you both sort of spoke when I listened to your speaking, I thought it was sort of interesting, like I think young people need to understand that, a lot of the things you mentioned were sort of basic science applications and mathematics and basic science and understanding some of these things. And the other thing as I was listening to you guys talk is for everybody out there, these are a couple of cow guys. These are a couple of cow people. These are ruminant nutritionists. And look at the way they're talking about the approach to their science, right? And how even ruminant nutrition has evolved and is going to evolve. And the other thing I thought about is listening to you, particularly for the young people listening, is that these two individuals, and I know you're very humble people, and you'll say, well, there's been a lot of people involved, and we'll say that too. There's been a lot of people involved. But this group has been a big part of the effort to reduce methane production in cattle. And 30 to 40% is a huge number. It's a huge number. And all the industries need to be looking at these huge numbers to reduce greenhouse gas emissions. And I'm very confident, I think, as I talk to you two, that the next molecules will be discovered and the next molecules will be effective. It's a great era we're in. So the last two, I'll just mention this and I'll let you guys make any concluding comments you'd like to make. The last two papers in this issue, one is by Auguste Del Prado, and he talks about sort of accounting, because it's really important to have accurate accounting of how much you're able to reduce methane emissions. Because if your accounting is bad, then you have no trust, right? You need to, and he does an outstanding job of just going through the systems to measure and account, how big of an impact are we actually having? Because initially there's so much methane produced by other industries and other activities that don't anybody expect that all of a sudden global warming is going to go down as soon as we start feeding 3NOP, because we got a lot of things we need to deal with. So you need to have accurate accounting, and he does a fantastic job of sort of summarizing that. And then of course the last article is we already talked about on sort of the different regulatory frameworks. So again, I would encourage those who are of interest to maybe, you guys have done such a great service, and I know there's many authors on this, but it's been a great service to the entire dairy and ruminant community, by publishing this. And when I looked at it, I was like, wow, this is fantastic. Are there any other, Andre, I'll let you start and then we'll let David finish, but are there any other things you'd like to leave people with about this issue or your experiences? And, it's your show, anything.
I think if you just look 10 years backwards, I think most people, even my own colleagues, not nutritionists, are involved in this area of work, but others, they would say, where's the catch? This will not work. And now you don't hear those sounds anymore because people see how fast the development actually has been. So I agree with you. It's quite astonishing what has been done the last 10 years. It's already in our farm assessment tools, the effect of 3NOP and of other additives, and it will be more. So I think there's big opportunities there for the next decade to come. Yeah, I think the impact can be very large. And actually all of this big, fast development in this type of research, actually that was what was leading us to having this flagship and saying, okay, there's so much research work going on. We need to, let's sit together and come up with some guidelines of how to do it, because there's a need to formulate those guidelines. That was actually the reason why we started this project, because of all this fast development and all the progress being made. So for me, it's a kind of logical outcome of the positive outcomes of the last decade of research. That's the reason why it's now in Journal of Dairy Science. Yeah.
Yeah. And David?
Yeah, no, certainly. Well, thank you, Matt, for your words. I think we, as a group of scientists, we really enjoyed going through this process of preparing the 6 papers. Obviously, with the help of, I'd like to mention Florencia Garcia, who was the postdoc employed in the project and funded by the Global Dairy Platform. She was great in helping us to coordinate the project. And as Andres said, this is just a result of what we learned in the last 10, 20 years. And we could see that the progress that the industry, the livestock sector has made is huge, but it could benefit from these kind of documents where the lessons that we learned in different areas of research are written in a series of papers, trying to cover the initial steps of trying to identify molecules to the very last steps on trying to account for the potential reductions, regional, national, or international levels. So it's just an exercise of putting together our experience, and hopefully it can help the livestock sector to improve the practices, and this is just only a contribution to reduce greenhouse gases emissions. Enteric methane is not the only contributor. Ruminants are not the ones to blame. It's just like any other sector in human activity. But this is just our experience, and we enjoyed writing it, and we hope it can be of help to others.
So I will conclude in just a second, but let me just state the obvious. Climate change is going to be one of the biggest challenges we face globally going forward. Science in this climate change area, which is what you guys are doing, I think it's fair to say you guys are in the climate change business. You're trying to mitigate greenhouse gas emissions. Going forward is going to be increasingly emphasized across nations. And if you're a young person and you have an interest in ruminant biology and ruminants in general, and you're in nutrition, let me recommend you look at this and read these articles, and it'll give you a start in this area of science. This is an area of science that is going to continue to grow, improving the efficiency of ruminant food production and also minimizing their impact on the environment. And what you guys have done with this special issue is given a whole new generation of scientists the opportunity to know what you guys know and take it further. And so I'd just like to thank you for your efforts. Thank you, Andre, and thank you, David. We're going to conclude here in just a second, but thank you for everything you guys have done for this thing.
You're welcome. It was a pleasure to work on these papers. Yeah.
Yeah. And thank you to Journal of Dairy Science for just taking these papers and accepting to publish a special issue. That was, I think it was very special for us as well to be able to publish it in Journal of Dairy Science. So thank you as well.
So thank you for that. To remind everybody, we've been talking about a special issue, Feed Additives for Methane Mitigation, is coming in January 2025. It's in the Journal of Dairy Science. This is 100% open access and free to anyone listening to this podcast or not listening to the podcast, free to anyone. And that brings us to the end of our show. If you enjoyed Dairy Digressions, be sure to like, subscribe, rate us, and spread the word to a friend or colleague. If you had feedback for us, please let us know at adsa@adsa.org for show notes and more episodes of Dairy Digressions, and learn about everything going on at the ADSA. Go to adsa.org. Until next time, thank you to everyone for listening, and thank you, Andre and David.
Thank you very much.